Yeah, can you see the PPT? Yeah, if you have any problem with like if my voice is not audible or if my slides are not moving, you just notify me, okay? So first we will discuss about various biomolecules. Then we will go into uh, the what you call digestive system and all these things, physiology, okay? So first biomolecules we'll be discussing today is their lipids. Okay, so what are lipids? So the word lipids has come from Greek term that is lipos. Lipo means fat. Okay. So here usually oils, fats, all these waxes, everything they come under lipids. Okay, occurring organic substances. So heterogeneous class means mixed, not only single type, but different types. Like as I said, oils, waxes, they are not same. Okay, so like that, we have different types of compounds which come under these biomolecules. And they have distinguished functional groups. They have different functional groups. For example, if you see cholesterol, you will have different functional group. You have a wax, so you will have different means different functional groups, different number of ring structures. So like that, there is difference between different lipids, and they are insoluble in water. Okay, so like all these examples, no, oil, is it soluble in water? No. Then wax, it is also not soluble. Even cholesterol, fat. So they are not soluble in water, but they are soluble in organic solvents like ether, chloroform, benzene, acetone. Okay, so this property of insolubility in water and solubility in organic solvents, that is only seen in the biomolecules lipids. So we have different types of biomolecules like carbohydrates, proteins, nucleic acids, and lipids. So all the other three, like carbohydrates or proteins or lipids, nucleic acids, they are all polar. That means they are soluble in water. But here only lipids are insoluble in water. And they are widely distributed in biological world. That means you can see lipids in every cell. So you know plasma membrane. So plasma membrane is made up of lipid bilayer. Okay. So not only this, if you see either plants, animals, microorganisms, or higher animals like human beings, in every organism you can see these lipids in one or the other forms. Okay. Suppose if you see in the honeybees, so they produce bee wax. So that is a lipid. And if you see lotus leaf, it will have a layer, okay, which will protect it. Like if you add a drop of water onto the lotus leaf, it won't, um, like it cannot stick. It will be non-sticky. So that is because of the wax present on that. So in that way, you can see these lipids in all the different organisms and all over the world. And they play important role in plant and animal tissue, as I said. So what are the main functions of the lipids? So lipids are mainly, they are the source of energy. Okay, 1 gram of fat gives 9 calories, kilocalories. That is 9,000 calories of energy. Okay, so now if you see, if we include like oils or ghee or anything in the diet, so nowadays like people say, okay, you will gain weight or you'll become fat, you should not eat. No, that is not the thing. So whatever the food we are taking, so we must include lipids, but in a certain proportion. So even carbohydrates or proteins or lipids, everything should be in certain proportion. So whatever the lipid we are taking, that will be in a stored form. So whenever we do some fasting, or whenever we starve. Okay, when this fat which is stored, that will be converted into energy and that will give us energy. Okay, so usually in the adipose tissue, this fat will be accumulated. So even not only as a form of energy, but in other functions also, like in the brain, in certain regions, we require these lipids. Okay, so it serves as a cushion for the vital organs and protect them from the external shocks and injuries. So this fat or lipids, they act as a cushion. So whenever we fall or some external injury, or like sometimes what happens, so in the brain, certain shocks we observe. So due to some unexpected consequences. Okay. So during that time, so to protect from the damage, these lipids act as a cushion. And they are structural membranes of the cells and membranes. Okay, structural materials like plasma membranes and even the myelin sheath of neuron. So that is made up of lipids. So cholesterol gives the flexibility to the plasma membrane. So all these are lipids. They are insulator for our body. Insulator means which will protect from the heat, means temperature changes. So see, our body, uh, it responds in a way to the temperature outside. Now if you see, this is winter season, so it will be very cold. So our body has to produce more heat from inside. If it is summer, what our body will do, it will produce more sweat so that it will cools down the body. So like that, our body will respond to external temperature and they will maintain the temperature. So during this period, suppose like polar bears and fish which live in polar regions, okay, poles, North Pole, South Pole. So those animals, they continuously need insulation from the cold which is outside. So for them, these lipids, they act as a insulator. Okay. Then lipids act as a carrier or reservoir for the fat soluble vitamins. So we have two classification of vitamins, water soluble and fat soluble. So for these fat soluble vitamins, that is ADEK. So they will get solubilized in the lipids. So therefore these lipids will act as a carriers for this fat soluble vitamin. And in food preparations, they also act as a binding agent, enhances the palatability of the foods. So these are the major functions of the lipids. Now we will look into the classification of the lipids. So mainly lipids are the two types: storage lipids, membrane lipids. So first we will see the storage lipids. So they are triacyl glycerols. So the name itself, tri means 3 acyl glycerols. So to the glycerol molecule, there are 3 fatty acid chains which are linked. Okay, these triacyl glycerols are usually storage lipids. Their function is to store the energy. Then membrane lipids are two types. These are present in the plasma membrane. Excuse me. So these are two types: phospholipids, glycolipids. So phospholipids are nothing but, again, there are two types: glycerophospholipids, sphingolipids. So phospholipids means a phosphate moiety is linked to the lipid molecule. So that is the name. So glycerophospholipids means to the glycerol, as I said, now we have already seen triacyl glycerol structure. So the same thing here, like instead of 3 fatty cell chains, here we have 2 fatty cell chains. And third one is replaced by a phosphate moiety and an alcohol. Okay, therefore they are called glycerophospholipids. So in the sphingolipids, we have instead of glycerol, sphingosine molecule. to which one fatty acid chain is there and one phosphate moiety which is linked to choline. Okay. So again, when we discuss like plasma membrane, so we have various functional groups like choline. They're called head groups. Okay. Choline, ethylamine, serine, all these, they are the amino acids, they link to the phosphate. So here, choline is linked to phosphate moiety of the sphingosine. They are called sphingolipids. Then, glycolipids. Glyco means sugar. Like carbohydrate moiety, when linked to lipids, they are called glycolipids. So here, these sphingolipids. So sphingosine, which have fatty acid, but here the third carbon, it is not having phosphate and choline, but it is having monosaccharide or oligosaccharide. Saccharide means sugar moiety. Okay, so that's why they are also called sphingolipids, but they come under glycolipids. Okay, so this is the classification of the lipids. So first we will discuss fats and oils. So fats and oils, they are the storage form of the energy in the living organism. As I told you, 1 gram of fat gives 9 kilocalories of energy. And they are the derivatives of the fatty acids. And what are these fatty acids? They are hydrocarbon derivatives, means or they are a chain of hydrogen and carbon moieties which are linked together in a certain pattern. So they are called hydrocarbon derivatives. They are long chain carboxylic acids. Carboxylic acid, COO minus, that functional group will be there. So up to 24 carbon atoms you can see. Okay. It can be 2, 4, 8 like that. Okay. 13, 15, anything up to 24 carbon atoms. So they are commonly seen in plants and animals. They are even number and odd number also. So even number are C16. So we indicate it as C16 means 16 carbon. C18. So C16 means the name it is palmitate. C18 stearate. Okay. Then fatty acids are classified as saturated and unsaturated. So saturated fatty acids means, so these long chain hydrocarboxylic moieties, chains, when they have only single bonds, they are called as saturated fatty acids. Only single bonds, no double bond. Whereas in the unsaturated fatty acids, you will have one or more double bonds. They are called unsaturated fatty acids. That you have to remember. So here you can see palmitate, stearate. So they are carboxylic acids, I said, COO minus. So that is at the functional group is at the top, first carbon. So then you have CH2, CH2 like that. You have n number of chain. So C16 and C18, I told you. So there is no double bond. You should not consider this double bond in the functional group. Okay, not COO minus, not that double bond. But in the chain, long chain of fatty acid, you should not contain any double bond. Then that is called as saturated fatty acid. If you see the unsaturated fatty acids, so here you have double bonds at the middle. Okay, so in the oleate, you have one double bond, whereas linoleate, you have two double bonds, right? So these, wherever you have double bonds, so they are called as unsaturated fatty acids. So one more important concept here is, if you see the first fatty acid, so this is the representation, how we represent. You have a blue color one that is called head group, and this yellow color one is tail. So this, whenever it is straight, okay, straight in the sense it is not rigid, it is flexible, but there is no bending, it is straight. So such kind of fatty acids are saturated. That means only single bond are present. Whereas the other adjacent fatty acid chains, wherever you see a bend, right? So they are unsaturated fatty acids. So wherever there is a double bond, there will be a bending. You will see. Okay. As the number of double bonds increases, the number of bendings also increases. Okay. So whenever there is a bending in between, the space between the two fatty acids, adjacent fatty acid chains, that will be increasing because of bending, there will be a gap. Okay, so you can see a tight packing only when there is a saturated fatty acids. That means whenever you have fatty acids with single chain or single bonds, so such kind of fatty acids are present, then all these fatty acids are tightly packed. If you have unsaturated fatty acids, then you can observe gaps. Okay. So this is how we represent. So this carbon skeleton 12 is to 0 means no double bonds. Okay, that is represent. So 12 carbons, you don't have any double bond. So this is the structure. Its name is do-decanoic acid. Decanoic, deca means 10, do-decanoic, 10 plus 2, 12, right? 12 carbons. Then its common name is lauric acid. So all these melting point, solubility, all these are listed. Like that, likewise 14 is to 0, 16 is to 0, 18 is to 0, means 14 carbons with no double bond, 16 carbons no double bond, 18 carbons no double bond, 20 carbons, like that. So below, if you see, 16 is to 1, delta 9. So that
Excuse
means,
me, ma'am.
yeah.
Ma'am, from which chapter you are explaining, ma'am?
So it's third unit, I think, last.
Last, third
I
unit.
don't, I don't remember the unit. You can just check the syllabus once.
Yes, ma'am. Like you're explaining from which your block, I'm not able to understand, ma'am.
You have the topic lipids.
Lipids we are having, ma'am. It is digestion of lipids. That's it, ma'am.
No, at the end you have biochemistry topic. No,
Yes,
it is
ma'am.
in biochemistry, right?
Uh, in biochemistry we are having that.
Yeah.
I will see once, ma'am.
Okay. So the last topic, I think, I don't exactly remember the unit name and all. Okay. So should I wait or shall I continue?
You can continue.
Okay. So if you see here, so 16 is to 1, 18 is to 1, like that. So 16 is to 1, delta 9 means that fatty acid is having 16 carbons with one double bond. That too, delta 9 means on the 9th carbon. Okay, between 9th and 10th carbon, you have double bond. So like that, the numbering will be given. So, even the orientation of the hydrogens also will be given as cis or trans. So no need to go in depth. But this is how the representation will be given. So 18 is to 1. 18 is to 2 means at 9th position and 12th position. 18 is to 3 means 9th, 12th and 15th carbon. So like that it will be given. Okay. So here you can see. So 9th position, how the double bond is there. 9th and 12th. 9th, 12th and 15th. There are 3 double bonds. So like this, they will give the numbering. Next, triglycerides. So they are the fats and oils which are found in animals and plants. So they are the esters of the fatty acids and glycerol. I told you, to the glycerol molecule, you have 3 fatty acid chains. They are simple lipids and they are storage lipids in the human body. So, so as I told you, this is a structure, 3 fatty acids and 1 glycerol moiety. Like this. You have a glycerol moiety to which these 3 OH, they are replaced by the fatty acid chains. So CH2, CH, CH2 will remain same, but These hydrogens will be removed, and to the oxygen we'll have that fatty acid chains. So like this, okay, 3 fatty acid chains. So this fat molecules, so same structure in detail, like with the example of fatty acid chains. So what are the types of triglycerides? So simple triglycerides and mixed triglycerides. So simple triglycerides are the triesters made from glycerol and 3 molecules of one kind of fatty acids. That means all these 3 fatty acid chains which are linked to the glycerol molecule, all these are the same type of fatty acids. Suppose it is oleic acid. So all these 3 should be oleic acid, like that. Okay, that is very rare. You don't find such kind of fatty acids very commonly, triglycerides. Then we have mixed triglycerides. So they are triesters which are different fatty acid chains. So you have animal fats and vegetable oils. So they have mixed triglycerides. So if you see butter fat, they contain at least 14 different carboxylic acids. So one is oleic acid, one chain, one other is stearic acid. Like that, you will see mixed triglycerides. So what are the properties? So hydrogenation of fats and oils. So what is the difference between the fat and oil? So usually we say all the lipids in a under single heading, but everything has a difference. So fats are solid in room temperature, whereas oils are liquid in room temperature. So this is the process by which this liquid oil is converted to solid fat is called hydrogenation, where adding a hydrogen to some of the double bonds, they will make that oil which is in the liquid form to convert into solid form. So here the catalyst will be usually nickel. So this we can do it in the for the industrial purpose. Okay, not in the body. Okay. Then rancidity. Rancidity is nothing but they develop unpleasant odor or flavor when exposed to moist air at room temperature. So this is by the hydrolysis of ester bond or oxidation of double bond. And here, this is because of the bacterial contamination which is present in the airborne bacteria, which will produce lipase. So therefore, this lipase will digest the lipids. So therefore, we have to store this food material which are rich in lipids in the refrigerator. And the oxidative rancidity is nothing but the double bonds are ruptured due to exposure to warm air. Okay. Usually we see cookies or even the food, oily foods, we make snacks and all. After some days, you will get oily smell like that. So this is all because of this rancidity. Then saponification. This hydrolysis of fats or oils by boiling with sodium hydroxide is called as saponification. This is mainly for making the soaps. Okay. So glycerol is an important byproduct of saponification. So if you see any soap, so glycerol, the component will be added. So this is to make the skin smoother. Your glycerol, it will make the skin smoother. Okay. The next classification is waxes. So they are also lipid family. So they are the esters of long chain saturated and unsaturated fatty acids with alcohol. So they have low melting point, they are stable solids, and they appear in both plants and animals. They protect the surface of the leaves from water loss, as I told you, like usually in lotus leaves and the best examples. So they also protect the leaves from attack by microorganisms. Carnauba wax is a major ingredient of car wax and floor polish. They have isolated this wax from the leaves of South American palm tree. Then beeswax, okay, which is miracle palmitate, okay. Then so like that, from both organism like animals, insects, and plants, you can extract this waxes. So the waxes also coat skin, hair, and feathers. They make them waterproof, okay. So here you can see the bee wax. So wax is an old English term. Weeks means the material of the honeycomb. So this is about storage lipids. Now we come to structural lipids. So the chemical analysis of isolated material shows that lipids are major components of most membranes. So they are not triglycerides, but another group of lipids are present, which are complex lipids. So triglycerides are the simple lipids, whereas other group are complex lipids. So there are two types of complex lipids, phospholipids and glycolipids. So phospholipids, they are the esters of phosphoric acids. There are two types of phospholipids in cellular membrane. So phosphoglycerides and sphingomyelin. So phosphoglycerides are also known as phosphatidylcholine. Okay. Apart from these, you have phosphatidylethanolamine, phosphatidylserine, phosphatidylglycerol. Like that, you have many. Okay. So, but here only one was given, like phosphatidylcholine, which is also called as lecithin. They built the long chain fatty acid, glycerol, and phosphoric acid. I have already shown the structure also in the previous slide. Then sphingomyelins. So here this phosphatidylcholine, they are present mainly in the plasma membrane. Okay. Then sphingomyelins, they do not contain glycerol. Instead, they contain sphingosine. Okay, long chain unsaturated amino alcohol. It is the only one fatty acid which is attached to the sphingosine. So these sphingomyelins are mainly seen in the myelin sheath of the brain. Okay, nervous tissue. They protect the nerves from the damage. This is phosphatidylcholine. So you have this is the glycerol. You have phosphate and choline linked. To the glycerol, you have two fatty acid chains. So to this phosphate group at the top, red one, and the choline. So these are the glycerophospholipids. I told you, like phosphatidylcholine. If choline is present, it is phosphatidylcholine. If ethanolamine is present, phosphatidylethanolamine. If glycerol is present, phosphatidylglycerol. If serine is present, phosphatidylserine. Other than that, other structure is same. Okay, these are all the components of the plasma membrane. Then glycolipids. Glycolipids are nothing but the glyco means I told you carbohydrate moiety, which is linked to lipids. It's called glycolipids, a lipid molecule that contains carbohydrates like simple sugars, glucose or galactose. They are also called as cerebrosides, which are mainly present in the brain. Then, where does these lipids present? So they are lipid bilayer. So majorly the plasma membrane which is present, that is lipid bilayer. So when you shake this phosphatidylcholine, you just take some phosphatidylcholine, add it to the water, and you just shake it vigorously and see under the microscope. It will form a liposome. Liposome is nothing but that it will form a circular structure where you will have these lipid moieties, that is non-polar moieties at the center and the polar molecules at the top, which is exposed to the exterior. So are you getting my point? So first you have to take some phosphatidylcholine, add to water and shake it vigorously. After some time, if you see, just imagine it will form a circle, closed circle. At the center, you will have water, right? And at the outside also you will have water. but at the middle you don't have any water. So wherever water is exposed, the polar substance will be exposed, whereas wherever don't see any water, there you will see the non-polar region. So that kind of structure is called as liposome. Okay. So it's a bilayer and it is thick wall of phosphatidylcholine. So here you can see the structure. So the round ones you can see like balls, white color ones, they are hydrophilic heads, and the two strands which are linked to each ball, they are fatty acid chains which are hydrophobic region. This is called liposome. So the same thing is seen in the form of a layer. Okay. So these phospholipids present in the water, they will have hydrophilic heads which are in interaction with water, and hydrophobic tails which hide from the water, and they self-assemble themselves as the bubbles. Okay. Then next are steroids. So cholesterol, then sex hormones. So steroids, now you see medicines, steroids will be given. All these are the derivatives of the steroids. Okay. Then, so there are 4 fused carbon rings. So they'll have different functional groups. With the structure which was given here is cholesterol. So, so cholesterol, it has a 4-ring structure and you have different functional groups. So in the same way, you have sitosterol, stigmasterol, which are present in the plants. So hopanoids, which are present in bacteria. Like that, you can see their different structures. Then, where are these sterols present? So the important part of sex hormones like testosterone, they are made up of steroids. Then with vitamin D, bile, bile fluid which aids in the fat digestion. Then adrenal hormones like cortisol and cholesterol, which is present in the foods made by the liver. Then dietary sources like in egg yolk, liver, meats, dairy products. So in that you can see the cholesterol. So this is the cholesterol, which is a very important part of the cellular component. So even in the plasma membrane, you will see this cholesterol. So in the animal cell membrane, so it will give the flexibility to the plasma membrane. Then it is the precursor for all other steroids, including vertebrate sex hormones. So they are high present in the high levels in the blood, and they may also contribute, like if we consume more meat or more cholesterol rich foods, and there is no proper exercise or that way, so the levels will increase in the blood and can cause cardiovascular diseases like heart diseases, like they will block the blood vessels, therefore the blood circulation will get affected. So they are the important components of the cell membrane. They maintain the fluidity and flexibility of the plasma membrane. So from cholesterol, sex hormones like cortisol and aldosterone, testosterone, estradiol, progesterone, and all these are produced. Okay, so they're all derivatives of the sterol or steroids. So now fat digestion. So how the digestion of the fat takes place. So now first, why I am telling you before starting digestion and all these things, why I am telling you about these fats or lipids is, so first you should know the structure. Directly if I say it is breaking in the double bond, you don't understand, right? So now you know the basic structure. You will have, if you see triglyceride molecules, so you have a glycerol molecule and 3 fatty acyl chains. So whenever these bonds are weak, so these hydrogen bonds, so these molecules will be first cleaved. So then it will form monoglyceride. Then the monoglyceride will be digested. Okay, so like this. So first carbon and third carbon, the chains will be cleaved. Then you will have monoglyceride plus 2 fatty acids. First, I will just tell you here. So the triglycerides and two molecules of water are split, and now you are leaving with the monoglyceride. This monoglyceride along with two fatty acids, they enters into the intestinal cell, intestines. Sometimes even the monoglyceride is also split into third water molecule. Then you will have three fatty acid chains and one glycerol molecule, right? So all these fatty acids, monoglycerides and glycerols, everything will be absorbed in the intestinal cells. So once they enter into the intestine, this first it enters into the small intestine. So in the small intestine, we have pancreatic lipases, intestinal lipases. So the lipases which are present in the pancreas and lipases present in the intestines, they act on these molecules. And once they enter into the small intestine, after the lipase digestion, then you will have bile fluid which is present. So that results in the emulsification of these fats. So here you can see. So how the emulsification of fats takes place. So you have gastrointestinal juices and water, and you have lipase enzymes. These red color ones are lipases. So whenever the fat molecule comes, this yellow color one, so this bile will result in the emulsification. That means it will catch hold of all these fat molecules. When fat enters the small intestine, the gallbladder secretes the bile. So this bile have both affinity for fat as well as molecules. So they bring together all fat molecules and emulsification takes place. Now this bile emulsification, it converts the large fat globules into small droplets to repel each other. Now this emulsified fat will be catch hold by the enzymes, and now each fat molecule will be digested by these enzymes which are lipases. Okay, this is by emulsification of fat. So now what are the functions of the fat? So in foods, they provide energy, they provide flavor, aroma and tenderness, they provide satiety, and they carry fat soluble vitamins, and they are the source of essential fatty acids. Okay.
Ma'am, what
Yeah,
is
yeah.
satiety?
Sorry.
The third point, ma'am, provide satiety.
Yeah, yeah. Just I will check you and let you know. Okay.
Okay.
Okay. Then, okay, so we have essential and non-essential fatty acids. So essential fatty acids are nothing but two polyunsaturated fatty acids. So essential means which should be in the digestion, like which is not provided in the body. Okay, so it should be supplied externally. Okay, so that's why like we have amino acids also, like essential amino acids, like essential carbohydrates, all these. Usually carbohydrates, they will be provided in the food, but here amino acids and all these things, they have to be supplied in the food. Some will be produced by the body through metabolic pathways. Okay, so sorry, any question?
No, ma'am.
Okay, so those fatty acids which should be supplied in the food, they are called as essential fatty acids. Okay, so two polyunsaturated fatty acids that must be provided in the diet are linoleic acid, linolenic acid. So linoleic acid are nothing but the other name is omega 6 fatty acids, and linolenic acid is omega 3 fatty acid. Okay, so nowadays if you see like ads, even like Horlicks and all, DHA, all these. So they are nothing but the essential fatty acids. So omega-3 fatty acids, if you see in the cod liver oil, fish liver oil, or you see walnuts. So nowadays people are aware that we should consume that which is not present in the, they cannot be synthesized in the body, and it should be supplied through diet, and that is essential for the brain development. So this linoleic acid and linolenic acid, so these two are the essential fatty acids. So they are found in the plant oils, nuts, seeds, whole grains, and fish. So they are important for the normal growth and development, and also they prevent the heart disease, hypertension, and arthritis, as well as cancer in different ways. Okay, so your question is here, satiety. Okay, satiety is nothing but it is like a sense of fulfillment, like complete. Like if you take the what do you call fatty acid rich foods, oil foods, so you'll feel full, stomach full, right? So that is nothing but the satiety. Okay. Um. Okay. So this
Yes,
is
ma'am.
all. Oh, sorry.
Yes, ma'am. Understood.
Okay. Okay. Sense of satisfaction. Okay, like that. So it feels full. So this is all about the lipids. So if you have any doubts, you can ask. So, okay, we will continue with the next topic, that is carbohydrates. First, I will finish the biomolecules. So, whenever I am teaching the biomolecules, I will also try to complete this physiology part. So, now I think digestion of lipids is also covered here. So, now we have lipids. So, you got to know what are the different types of lipids. So, what are their functions? So, what is their structure and how they get digested? Okay, this is a basic understanding you might have got. So in the next class we will continue with the next biomolecule, that is carbohydrates. Okay.
Excuse me, ma'am.
Yeah, please. I cannot hear your voice. Your voice is breaking. You can type it in the
See
chat.
with MCQs for 20 marks, and we are supposed to answer end of the questions. And it's saying the question will be given based on the knowledge that you have regarding the.
Sorry, your voice is continuously breaking.
Ma'am, she is saying that our paper pattern has changed.
Okay.
So we are having first like MCQs we are having for 20 marks. Now we are having one question, like one answer for 20 marks.
Okay, okay.
So which question we can expect from that, like for
Oh,
20 marks?
okay, okay. So I understood. I will just give you the sample question later. So once I finished. Okay.
Okay,
Means
ma'am.
usually if you have any metabolisms, you can expect metabolisms. It is just you can expect. I am not sure. I'm not telling that you will definitely get it. Because you should estimate that 20 marks a single question means it is like an essay. So you
Yes,
should
ma'am.
have that much matter to write also, right?
Yes, yes.
So if you have any metabolisms, so you have to write the cycle, you have to explain the steps. So such kind of questions you can expect, obviously.
Okay, ma'am.
So,
Okay.
but anyway, I will just go through the complete syllabus. If I find any potential questions, I will just discuss in the next classes. Okay?
Okay, ma'am. Yes, ma'am.
Okay. Okay, fine. So I think no questions from today's class. So I am just ending up the class. Okay? Thank you so
Okay.
much. Yeah, thank you.
Thank you, ma'am. Thank you.